History of Plant Pathology Part 2. M. J. Berkeley, 1855. Observed root knot nematodes in galls of cucumber roots. Founder of Mycology in British described 6,000 species of fungi. Studied the late blight of potato pathogen, rust of coffee and grape downy mildew. Authored books. 1. Outlines of British Fungology. 2. Introduction to Cryptogamic Botany. Anton van Leeuwenhoek. He was a cloth merchant living in Delft, Holland. Developed magnifying lenses, 300 to 500 times magnification. He developed simple microscope. First to discover the microorganism like bacteria, protozoa, algae and named them as little animalcules. He is regarded as father of bacteriological world. Louis Pasteur proved the germ theory. Coined the terms aerobic and anaerobic organisms. Proved fermentation is biological process. Developed pasteurization process. Developed vaccine for rabies disease. Regarded as father of microbiology. Robert Koch, 1876. He proved that the anthrax disease of cattle is caused by a bacterium, Bacillus anthracis. He gave Koch's rules or Koch's postulates or proof of pathogenicity. Use of gelatin as a solidifying agent. Found causal organism of tuberculosis disease. He considered as father of medical bacteriology. Regarded as father of bacteriological techniques. R. H. Biffen, 1905. Resistance to yellow rust in wheat was inherited and follow Mendelian fashion. First to develop rust resistance variety rebet against yellow rust of wheat, Puccinia striformis. Flo, 1955. Given gene for gene hypothesis concept in flax against rust, Melampsora linni. For each gene confers resistance in the host there is a corresponding gene in the pathogen that confers a virulence and vice versa. Vanderplank, 1963. Given the concept horizontal and vertical resistance, threshold theorem, vertifolia effect, AUDPC, area under disease progress curve. He written books on 1. Disease resistance in plants, 1968. 2. Principles of plant infections, 1975. 3. Plant diseases, epidemics and control, 1963. Regarded as father of epidemiology. J. H. Craigie, 1927. Discovered the function of pycnospore of Puccinia graminis tritici. Discovered the sexual stage of rust fungi. Blaxley gave concept of heterothallism in fungi. J. C. Walker reported the role of phenolic compound in plant defense mechanism. Reported the resistance of colored onion bulbs to smudge fungus, Coletotrichum cercinans, was due to catechol and protocatechuic acid present in the dry scale. He wrote 2 books on Plant Pathology, 1968. Diseases of Vegetable Crops, 1952. Muller and Cruikshank. Involvement of phytolexin in plant disease resistance. Walder and Frau Hesse, 1881. Used agar as a solidifying agent in media. R. J. Petrie. Developed the petroplates to circular plates holding the media. Daw and Ishii, 1967. Discovered mycoplasma-like organisms, amylose, causing yellow stripe diseases, mulberry dwarf, aster yellows, in plants. Ishii et al. Reported tetracycline can be used for management. Davis and Worley, J.E., 1972. He first time discovered spiroplasma in corn stunt disease. Windsor, I.M. and Black, L.M., 1972. He observed first occurrence of rickettsia-like organisms, arillose, in clover club leaf disease. Virology. Carolus Clusius. Recorded breaking of tulips due to viral infection. Adolph, 1886. First time discovered the tobacco mosaic virus, TMV, disease. Named the symptom as mosaic. Showed that sap transmission of disease. He demonstrated that this disease, TMV, could be transmitted from disease to healthy plants by the sap from leaves showing mosaic symptoms. Dimitrii Ivanowski, 1892. He demonstrated that tobacco mosaic virus, TMV, could pass through even those filters that retained, to keep, bacterial cells. So he suggested that viruses were smaller than smallest known bacterium. M. W. Beijerinck, 1898. He concluded that tobacco mosaic virus, TMV, was caused by contagium vivum fluidum, infectious living fluid, which he called a virus. He discovered the causal agent of tobacco mosaic disease. Coined the term virus. Father of plant virology. Stanley, W. M., 1935. He purified and crystallized tobacco mosaic virus, TMV. Reported viruses are proteinaceous molecules. He got Nobel Prize. F. C. Borden and N. W. Peary, 1938. Proved that viruses were nucleoproteins. Showed that TMV consists of 95% protein and 5% nucleic acid. Jura and Saturam. Showed RNA of tobacco mosaic virus, TMV, is infectious, not the protein part. Knoll and Ruska, 1932. First invented electron microscope. Diana, T.O., 1971. Discovered the VIROIDS. Determined that potato spindle tuber disease is caused by small molecules of infectious ribonucleic acid, RNA, which he called VIROIDS.
Dear friends, if you like this video, then like, subscribe and share.